Okay, my friends, in this video, we're going to be talking about BUN, creatinine, and urine-specific gravity. And before we dive too much into that, I just wanted to take a quick moment and point out that the kidney is a super cool organ. It's the only one where diagnostically, we can look at what's going into it, so what we're getting out of our blood here as it goes into the organ, and what's coming out of it, the urine, and look and evaluate what the urine is doing. And between knowing what's going into it and what's coming out of it, we can infer quite a bit of how the kidney is functioning and what it's up to. So without further ado, we're going to pop in here and take a look at some of the things that we look at when we're looking at kidney function, but we're going to be focusing on BUN and creatinine and urine-specific gravity. So to start off with, a CBC, a complete blood count. So for those of you who haven't encountered this before, a complete blood count, it looks at all the sort of cellular material of our blood. So it includes things like white blood cell counts and all the different white blood cells that we see, our red blood cell count, and all the different parameters that go around red cells, as well as platelets. For the kidney, one of the things with kidney function is we pay particular attention to our red blood cells, remembering that the kidney produces erythropoietin, and that hormone stimulates red blood cell production at the level of the bone marrow. On our chemistry panel, though, this is where the real money makers happen here with the kidney. We can see here our urea nitrogen, which is another term for blood urea nitrogen, and creatinine. And this is what we're going to be focusing on today. But there's a whole bunch of other things in our chemistry panel that the kidney is involved in regulating. Things like our electrolytes and acid-base status, our calcium and phosphorus homeostasis, even glucose. We know that it resorbs it up in that proximal tubule. Albumin and total protein. This becomes important with glomerular health in particular, but there's all sorts of other things that can play along with that as well. But like I said, we're going to focus on these two for today, and we'll get into all the rest of these when you get up into your clinical pathology course about a year from now. So without further ado, blood urea nitrogen, or BUN, also known as urea nitrogen or serum urea nitrogen. You may hear all of these terms and we'll see them utilized around. This particular analyte, where does it come from? Our blood urea nitrogen comes from proteins in the small intestine. So we know that proteins come in, they're broken down into their amino acid pieces and absorbed into the bloodstream. And the liver takes these amino acids and it plucks off the amine group and shuttles it into the urea cycle. Urea cycle, aptly named, as it finishes up with producing urea nitrogen, which is then moved into the blood, which is where we get our blood urea nitrogen terminology, or BUN. In the blood, a little bit's resorbed back into the gut, but most of it is excreted in the urine. So it passes freely through the glomerulus, a marker of GFR then. And the one thing about it is a lot of it is resorbed. So BUN tends to be resorbed back into the blood and only a little bit of it excreted into the urine. So it's an okay marker of GFR, but there's a lot involved in it. So we need to know how much protein an animal is making, how that liver is functioning, what the flow rate is through the kidney itself, in terms of how much is being resorbed or allowed to be resorbed, and it's multifactorial. In our large animals, it gets even more complicated. In ruminants and equids, in particular, they excrete BUN not only through the kidney, but through the GI tract is a major source of excretion. And what ends up happening, our ruminants are particularly good at this, so we're just going to use them as the example. BUN gets excreted into the saliva, goes to the rumen, and the rumen microflora there take it and make it back into amino acids. And so what ends up happening is that urea ends up being lost and proteins are made. So we have a net protein gain and BUN loss. This makes it an even more unreliable marker of kidney disease. So in our large animals, we concentrate a lot on creatinine in particular and urine-specific gravity, and we put a lot less emphasis on BUN. A little bit more in our small animals. Creatinine is sort of our go-to, and I know you've encountered this in physiology already. Creatinine is an excellent indicator of GFR. So where does creatinine come from? It comes from muscle. So it is a byproduct of muscle metabolism. So creatinine phosphate, that phosphate pops on and off to make energy, and creatine and creatine phosphate will both denature into creatinine. That creatinine will move into the blood and be filtered at the level of the glomerulus, where almost all of it's excreted. I know it's not a perfect 100%, but biologically speaking, when we're thinking of it in a medical term, we think about it in more simplified big picture terms. The one catch about creatinine is that it is dependent upon muscle mass. So animals that are highly muscular will have normally higher endogenous creatinine levels, and animals that are muscle wasted, like our old cats in chronic renal failure that tend to be really thin, won't have as high of a normal creatinine, and so it can mask the severity of kidney disease. And that's the challenge with this. So those of you who may have heard of SDMA, we're not going to talk about it here. We will talk about it when you're up in clinical pathology as an alternate marker that's hitting the market these days that helps pick up kidney disease a little bit earlier. This one is, SDMA is not dependent on muscle mass, and that's sort of its big marketing strength. So alongside our blood, so we've got our BUN and creatinine, we've familiarized ourselves with that. We are going to be looking at a urinalysis. And the thing that we're going to be focusing on today is our urine-specific gravity and what that means, and talk about that in terms of the medical sense. There's all sorts of other things that urinalyses show us in terms of what the kidney is producing and what's present in there. But for kidney function in particular, we're going to talk about urine-specific gravity today. So what is urine-specific gravity? Urine-specific gravity is a reflection of the kidney's ability to concentrate and dilute urine. So we know that the kidney resorbs water, and in general, we operate in a mild state of dehydration, meaning that the kidney is normally resorbing water. It's part of its normal function. We have this area here, which is our loop of Henle and the concentration gradient that develops here, that's dependent upon urea or BUN production. So we need a nice functioning liver to keep that up and going, as well as production of aldosterone, which helps us resorb sodium and keep that gradient nice and dense, so water will move in here and have some passive resorption there. We also see resorption of water here at the level of the collecting duct, which is where our friend antidiuretic hormone, or vasopressin is the other term that's used, operates here in terms of expression of aquaporins and drawing of water back into the interstitium. So in our kidney, we need functioning at both of these levels to have concentrated urine. So it's one of the things that we keep a close eye on. When we're talking about measuring our urine-specific gravity, we measure it with a refractometer. If you haven't encountered one of these yet, you will a lot in your veterinary career. The basis of this is we put a little drop of urine on this piece of glass here and pop down the top part, and we have an ocular here, and we take that ocular and we peer up into the light. And what we see on the inside of this ocular is our grids. And depending upon how well concentrated that urine is, it will give us an idea, a measurement. We'll see a blue line come across the screen. And where that level of the blue line is gives us its specific gravity. So most refractometers measure from 1.000, which is deionized water, pure water, up to 1040. Okay, and this is the terminology that we use in medicine for specific gravity. I know it's 1.040, but the shorthand that we talk about it is 10 and then the number. So 1040, 1080, 1001, 1055. Just be careful knowing that decimal place falls right after that one. It takes a little getting used to, but that's the lingo. So most refractometers measure up to 1040, and if it's greater than that, we know it's super well concentrated. Some of them will go up a little bit higher than that. The long and the short of this is the higher the urine specific gravity, so the farther away we get from a flat one here, the more concentrated the urine is. And knowing that kidneys are supposed to resorb water and concentrate urine, that's part of what they do, we look to make sure that that's functioning and happening. Keep in mind, there is some range of urine-specific gravity by species. We're not going to spend too much time on this, but suffice it to say that cats, being desert creatures, are absolute ninjas at resorbing water. Their urine-specific gravity can be up to 1080. Dogs, close second at 1060, and our horses and bovids go up to about 1055. A little variation. Don't sweat this too much. You can sweat it when we get up into clinical pathology, where we'll emphasize that again a little bit more. So our descriptors for urine-specific gravity. When we are talking about this, we've talked about the number system and how we talk about 1040, 1030, et cetera, et cetera. There's different categories that urine-specific gravity falls under. And it basically focuses around this green spot right here. So this is isocinuria, and this is the range of 1008 to 1012, okay? And in this range, the urine is at the same level of concentration that the plasma is. And so what it means when we get a number in this range, it means that the kidney is not adjusting the urine concentration. It's not adding water to it, and it's not taking water away. So this is sort of the one that we put our eyeballs on when we're concerned about kidney failure, because if the kidney is not functioning correctly, it's not going to be able to adjust its urine concentration. From this point up towards the more concentrated ranges, we have a couple of different ranges. One is the range of minimal concentration. Varies a little bit by species as to how well they concentrate their urine here, okay? But this is sort of gray zone. There's a bit of urine concentration going on. It's not super concentrated. And then this top part here, over about 1030, 1025 for our large animals and 1035 for our pussy cats, is what we call concentrated urine. So well-concentrated urine, the other term that's used is hypersthenuria. You may hear that thrown around once in a while, but mostly we say that, wow, it's really well concentrated when it's up there. When we've got nicely concentrated urine, we're going, kidney's working, it's doing a good job. Range of the minimal concentration, most animals, and just on a normal day-to-day basis, will have urine that's over 1020 in terms of being concentrated, okay? Most of the time, they're even over up in this well-concentrated range, but you never know when you catch them with their drinking levels. Especially it's in the middle of the day or the evening, you don't know if they've been at the water bowl recently, so it can sometimes vary during the course of the day. The last sort of spot here is hyposthenuria, and we're not going to talk about this because too much. The kidney's functioning in this range. It's just in this range, it's adding more water to the urine, not necessarily concentrating it, but diluting it. Threw it up here just for your edification at this juncture. All right, which brings us to the end of our talk. So the next part that we're going to get into is we're going to start talking about organizing azotemia and how we put BUN, creatinine, and urine-specific gravity together to figure out if the disease is at the level of the kidney, or if it is before the kidney or after the kidney.